You're listening to the Turing Podcast, a production of the Alan Turing Institute, the UK's National Institute for Data Science and Artificial Intelligence.
Hello everyone, and welcome to another episode of the Turing Podcast. This is another remote edition being recorded in the midst of coronavirus lockdown. And I'm your host, Ed Calstry, Junior Research Data Scientist at the Alan Turing Institute. Today I'm joined by Bea Costa-Gomez, who is a PhD student at the University of Manchester in the Faculty of Biology, Medicine and Health, who recently completed her Turing Enrichment Scheme placement. Hi Bea, how's it going?
Hello, everything is fine. Just enjoying the social distancing. Speaking. Yeah, exactly. I like how you specified coronavirus lockdown in case there's another lockdown, and this one is a very specific one.
Well, I hope not. That would not be that would not be ideal. I should say for our listeners, we're recording on the 21st of April, so if the world has plunged into chaos further since then.
Yes, if there's zombies around, at this point it's only coronavirus still, so that's fine.
Yeah. So it's a relatively easy lockdown by comparison to what we've got coming.
Yes. So future selves, I'm sorry, I jinxed it. Now we'll see. Hopefully I didn't.
Mind you, in some ways this is worse, because if you've seen the film Shaun of the Dead...
I was thinking about that film exactly. I have
at least they get
zero
to go to
vinyls. I don't have any vinyls to throw in case zombies come and attack me, so I don't know what I'll do.
No, well, yeah, I was going to say at least they get to go to the pub, which we can't do at the moment. No. So, in a sense, coronavirus is worse than the zombie apocalypse.
There's no Winchester for anyone. That's it.
No Winchester. All right, Bea, I'm going to ask you a few questions about your PhD work in neurodegenerative disease. Perhaps before I start, do you want to just give a brief summary of what you've been working on, and specifically the kind of area of research that you worked on at the Turing Institute?
Yeah, so my PhD has completely derailed from its original plan, but the idea is that I'm studying neurodegenerative diseases. But particularly, I built a software that analyzes cell culture. Images of neurons in cell culture, which means that I have isolated neurons, which are brain cells, and they, with certain genetic changes, they actually have these symptoms that appear in a range of them, and a range of diseases, and I'm interested in studying these. And so I built a software, which is the Advanced Labeling, Fitting, Recognition, and Enhancement of Data, or ALFRED, whatever you prefer to call it. And
that's a nice acronym.
Thank you. It's Alfred for, Batman's butler, so that you're Batman when you use the software. I have a lot of free time. I don't anymore, but I used to. Is
Alfred, there any significance to that choice?
To Alfred?
Yeah.
Can you imagine my senior supervisor saying, "How's Alfred? Is he okay?" And it's a software. I just enjoy it. I do it for that. It's just fun. And the icon is actually a little butler. A friend of mine did an 8-bit butler for me, so it's just fun. All of my laptops have also names of superheroes, so that's fun.
Nice.
But basically, Alfred, you load the images. It does all of the pre-processing that you need, and looks at things like curvature and other shape characteristics of the cells, because the diseases, the particular symptom that we're interested in, and this is going to be helpful for everyone hearing us, because even if I had images I couldn't show, right? But the cells, the neurons are, if you look at your arm, and you can imagine that your hand is the cell body, and then you have the arm is the axon. Which is actually the whole of the length of these cells. Imagine your arm. And they need to keep this structure for the entire lifetime. So they have something inside called a cytoskeleton, which just means skeleton of the cell. And the backbone of the skeleton it's called it's these polymers called microtubules, and microtubules normally look like just a bundle of dry spaghetti. So imagine dry spaghetti all over the length of your arm. In these diseases, sometimes you look at the axon and you see a swelling. You see like this bump, and when you look inside the swelling, instead of seeing the bundles of dry spaghetti, you actually see what it looks like a bowl of cooked spaghetti, which is very different, and you have these loops and turns. So we want to actually quantify, find a way of quantitatively compare these things. And because it has loops, that's why the curvature is interesting for us, and all of these shape characteristics that Alfred analyzes. And then
to back up for a second, it's what kind of neurodegenerative diseases are there that make the neurons, which are normally like spaghetti, change into this different shape?
So it's diseases mostly for peripheral neurons. So motor neuron disease and Charcot-Marie-Tooth is another one that has it. So these are not very specific, but it's a common symptom in diseased cells and aging as well. This also happens in normal aging of neurons. So sometimes they just has that. We don't exactly understand why that happens, and that's where we come in to study this.
Okay. So that leads me perfectly on to the next question, which is that image recognition is a hot topic in AI research, generally speaking. How does your research make use of advances in this field?
So that's why I actually went to the Turing for my enrichment scheme, because back in Manchester, here, where I am now, my supervisors, as I said, my PhD completely derailed from what it was supposed to be. So my supervisors are a biologist and a mathematician. And I realized that it would be all of this research that I was doing was based on the assumption that we made that the curvature was relevant to analyze these images. So I thought maybe I can actually apply some machine learning, because I did some of it during my master's, and I thought I can do that. So I actually applied for the enrichment scheme to have a collaboration potentially with someone from the Turing, and that's why I met Dr. Ian Stiles from Birmingham, and I collaborated with him. And so image recognition is a very hot topic, and there's loads of it. And what I use two types of things in this in my project. So I use cell segmentation, which is it's basically a subset of image recognition where it looks at a cell, because cell cultures usually are a black background with colorful things on top. And this is a semantic classification generally of cell, not cell, or cell 1, cell 2, and then not cell. That's the first part that I use on my project.
So is this a commonly used kind of image recognition algorithm for distinguishing between biological samples or cells?
Segmentation is something that happens very, very in all sorts of applications, because these are that's the first step to analyze biological data. You need to actually tell what is the interesting part in the whole image, because you're going to have usually a lot of noise in the image that is not relevant.
Okay. So in your case, the interesting parts in the images, when you're looking at images, I guess, of diseased neurons and non-diseased neurons, is something to do with the curvature of the axons. So was that something that's been known for a long time, or is it something that's come about from the analysis of these images in your own research or in previous people's research?
It came out from the research in my group, actually, from the biological perspective of it. They've noticed that there were these swellings, and they came up with a theory to explain. It's called the local axon homeostasis, which is just a theory of how the proteins in the axon actually make the disease. So the curvature is it's because it's a very obvious thing when you look at the images, because just think about dry spaghetti and cooked spaghetti and how conformational differences are. And if you have, and you don't have basically almost any curvature in dry spaghetti, right? It's very stiff rod. And with cooked spaghetti, you have all of this. So that's why curvature jumped to the eye, because it's a very obvious consequence of this disease.
And remind me again which way around it is, with it, whether it's the cooked spaghetti that's
the cooked spaghetti is the disease. Yeah, that we do not want cooked spaghetti.
Okay, so normally all of our neurons are these long straight. Well, they have these long straight parts which are called the axons. In these neurodegenerative diseases, it becomes spaghettified, like cooked spaghetti.
Yeah, it's like a swelling. So it's not all of it, but you just have like a swelling in the middle. Sometimes more than one. Just like imagine there's a lot of it going well, and then it goes wrong, and then it goes well again, and we just don't understand. But there's very obvious consequences for this, because so neurons communicate by electrical signals between each other, right? And the electrical signal goes all along the axon. So this is from physics. What's going to happen if you have a bump in the middle? It's going to delay the reaction, and it's going to.
I see.
Do you see what it means? Like if you have the swelling in the middle, then it goes bump, 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 slow down, and then that is.
So this is, you're kind of thinking that when it's interrupting the path of signals that would be our movement or our thought processes.
I have a good analogy for it. So just imagine a literal electric cable, and it has a knot in the middle, but inside the rubber. So you have the rubber is fine, and then you look at it, and it's like a swelling in the middle, and all of the electrical cables inside, all of the copper cables are tangled in the middle.
Right.
So that's what it looks like.
Okay. Well, so it sounds like the differences, especially from those images, are fairly clear. But how good is your software that you've developed, Alfred? How good is Alfred at categorizing those neurons between the diseased and non-diseased?
So between disease and non-disease, that's good. And the classifying algorithms as well, because also what I've done, the second part of my enrichment at the Turing was actually using a classifying algorithm, machine learning algorithm to just a neural network to classify disease, non-disease. Those are relatively easy to categorize. They are longer. The non-diseased are longer and are thinner, and the curvature doesn't change much. So the curvature along the axon is quite similar. However, what we're interested in is comparing. It's comparing between diseased neurons. So the point of the question of my project, actually, I should have mentioned this before, is that these diseases that have this symptom are very different diseases. So generally, you have a disease. What sometimes happens is that you have a symptom that shows up because you are altering the pathway of proteins, but they're all in the same line. It's like you have a factory and you just interrupt one of them and one of the workers, and then everything. The end product is different. What happens here is that you have the disease when you alter different non-communicating lines of production. So it was supposed to be two completely different things. So the ones that I study a lot are regarding proteins that help the guidance of the growing microtubules when they're growing. So helps the spaghetti being parallel. And also proteins that walk along the skeleton, that transport things inside the cell. So as you can see, they're very different things that interact with the skeleton, but give supposedly the same result. So what we want to know, and the big question of my PhD is, are these symptoms the same? Is it the cooked spaghetti that we see? Is it the same in the both diseases, or is it just similar? Because if it's the same, then we can say with more certainty that it's probably because of the microtubules themselves. But if it's different between them, then maybe it's something else that is happening that is perturbed by both of them and reacts differently to each. So the interesting thing is that that's why the machine learning came in, which is, are the images distinguishable between diseases by a machine? And are they distinguishable by non-machine, which is what Alfred does?
Okay, so I guess the question really is, because we know that the result of different diseases is a similar structure in terms of the neurons, ends up in a similar shape. But is there enough difference in that shape to distinguish between the diseases from the point of view of these machine learning algorithms? That kind of, well, I guess I can ask you, well, how well did it do?
Terribly.
Okay.
I did I tried a bunch of them. Yeah.
Yeah. This actually, the question I was going to ask next, which I think this is a good time to ask it, is: do we now think that the different neurodegenerative diseases are causing the neurons to be spaghetti, all the other way around? Because I guess if it was the other way around, we could figure out how to unspaghetti the neurons. That would potentially cure all of these diseases in one go.
That would be so nice.
And it said, but it's what you were saying to do. So do you think now it is to do with the microtubules themselves?
That is the ongoing discussion that I've been having with my supervisor for the past 3.5 years, because at some point the more I look at the images, the more confused I get. I just see these images everywhere now. I sleep, I dream about them, and now I'm writing my thesis, so you can imagine how exciting it is. But the truth is that we still don't know. I still need to do a lot of analysis, but it's still a big question mark. But the truth is that a product came out of the PhD, so I have the software that everyone can use to get the parameters out of their images. I usually in conferences, I presented a lot in biology conferences, and I get asked, does it only open images of neurons? And I say, no, you can open any image that you want. You can check whether Cheerios are actually all the same curvature that they promise that they are on your. So just take a picture of your breakfast and show it. But it's just the question is still there, and we're still investigating, and there's still experimental. Biological experiments being developed. Not anymore because the lab is closed, but I have a postdoc in the group, Dr. Andrei Volzman, and he basically is a magician in biology and amazing, and he comes up with all of these experiments and does all of the experimental part, and comes up with the images and ideas for us to test.
Cool. Well, I guess would you be confident at this stage at saying
No.
Hang on, you haven't heard what I'm going to say. The algorithms you have used and that form part of your software, so they haven't distinguished differences in the way that the different neurons from different degenerative diseases, or the differences in their curvature. So do you think that that's really because there is very little difference, and it's the different diseases are causing the same thing to happen to the neurons? Or do you think that there might be more subtle differences that your algorithms are yet to pick out?
So the only thing that we get from Alfred is curvature and straightness ratios and distributions. So what we need to do is actually go through all of the images and then at the end compare them. One of the main ideas is that we're going to use this as well. The algorithms that I've developed with Alfred are actually just pure maths retrieving the values from the images. So there's no machine learning in them. The point that we want to use is that I am training an algorithm to distinguish. I'm currently training a ResNet neural network to distinguish them, the diseases. If it works, some days it works, some days it doesn't. There's a very disturbed. It has its own personality at this point. That I don't understand.
It's got its own. It's got its own neurodegenerative disease.
Oh yeah. Oh, definitely. This algorithm needs therapy at least to see if it decides. Because if it decides to not work, that would be one thing. But some days it gives me a 40% accuracy, and other days a 70% accuracy, and then I just get a question mark on my face. But the point is that the idea is that also after using Alfred, instead of comparing both, I can actually join them together, because Alfred gets extra information about these images. So maybe I can feed that extra information. I can feed the curvature distribution and the straightness distribution and the length distribution of my neurons into the classifying algorithm, and maybe that could be also helpful to distinguish between diseases.
But what would be the ultimate goal then? If you're able to come up with a piece of software that can distinguish between these diseases, how would that help in the combating these diseases in the long run? How would that knowledge help?
So the way that would help, because we would understand what's going wrong, because currently we don't know. We are working with very basic biology. So this is a disease that has very obvious outside symptoms in people, but the truth is that we are looking at really, really, really detailed things that we still don't understand. So the idea of these pipelines, air quotes, you can't see the air quotes, but I did it. The idea of these pipelines is that you start by understanding the basic concepts of what's happening, and then if you understand the way it's supposed to work, then you can notice when it's not working well, and you can do something about it. Because these are genetic diseases, so it's not a disease that you can some of them are genetic, so it's not something you can prevent. But maybe if we understand why it goes wrong, it can be preventable in, for example, aging, and things like that. So it's a small step, very small step, but hopefully it can make a change.
Well, every little helps, doesn't it? This is how science progresses. You've got to go one step at a time.
And because I made a tool, it also means that people doing other types of research can actually look at it, because everything can be studied in terms of curvature. Like, for example, I've had people ask me for my software to analyze blood cells, because sometimes in some diseases, instead of having a very round red blood cell, you actually have like something that is a completely different shape. So the curvature distribution of those two shapes would be very different, theoretically. And so you could use that. You could use it to study movement patterns, because they are also the way the movement of, for example, larvae is recorded is that they have the center mass. Just imagine the center mass of a point moving around, and when it's diseased, it doesn't move as much. So you could do a curvature pattern of that, for example. So these are very different.
When you say larva, what do you mean?
Like fly larva. Like there are a lot of diseases that are actually studied into like those little slimy tiny things.
Well, I was going to ask you about that, actually. I remember when I attended a talk you gave about this research previously, you mentioned about the Drosophila flies. How do researchers in general use them for studying neurodegenerative diseases, and how, if at all, did this factor into your research?
Okay. I'll start with the second part. How does it factor into my research? Because I have a sentence that I enjoy saying very much, which is how my work is parallel to what my group does, because they smash the brains of the bugs to get the cells out, and I smash my brain cells to get the bugs out. So that's the essence of what we do. So as I said, I work with very basic. We work with very basic biology, and there's a lot of conserved mechanisms. I'm sorry if anyone is listening that doesn't believe in evolution. Sorry, this is the wrong podcast. But there's a lot of conserved mechanisms between flies and and humans actually.
So you're saying that I'm the same as a fly? I'm basically
that is 100% exactly what I said. Not even the conserved mechanism, but that's exactly. You're a fly, basically.
Well, I for one am offended, but please continue.
I was gonna say, I'm sorry to offend you guys. So I actually was reading about this because I was writing like the structure of my introduction where I need to go through all of this. So there's actually, I think it's 75
your PhD thesis,
yeah.
Is it right?
Yeah.
Right.
So much fun. I'm just enjoying. It's a thrill. This lockdown writing thesis.
Hey, you've got lots of time to.
I know.
Get on with it.
I know. Help me. Okay, so there's about, actually, I think it's 75% of diseases, human diseases that have a homolog gene. Homolog means that it's the same gene but in the fly. For basically what this means is that we have a very complex system, of course, and then what happens is that some of our diseases happen because there's a slight change in a gene in the middle of your big DNA, and there's an analog of that on a fly. And if you can find the analog of that on a fly, then you can study the changes that causes in such a small system. So it's this, imagine, for you computer scientists, imagine that you have a massive database and you have a small algorithm that you want to test. You're not going to test on the entire database. You're going to take a sample of the database and you're going to test it. So what we do is that there's a massive complex system, which is the humans, and we can actually sample this on the fly, on this very particular basic mechanism. So we just do it on the fly. Because, as you can imagine, people are not too keen if we go around smashing brains of, humans. They're not very happy with that.
You never know. I mean, if in a few months, as we predicted.
As I said, if a zombie apocalypse shows up, maybe we can study those brains and just go and smashing brains. That's not as effective. But you just go... And also, it's much cheaper. This is the Drosophila marketing. It's very cheap to keep flies compared to other model organisms. They are very easy to genetically manipulate. You can literally have a new generation in 3 weeks, which is something that you can't with humans. If you need to do some genetic manipulation, it's not as quick.
Takes a little bit longer.
Yeah, sci-fi makes it seem so much quicker, but it's not. No, just think of it wouldn't work that way. And even if you could genetically manipulate a human, it would take 9 months for the human to be born. When you can get so many generations of flies in that time. So again,
I can imagine as well that using them for this purpose as well, as you were saying earlier, you're studying genes that obviously have a deep evolutionary origin, and that kind of makes sense that even though a fly's brain is much smaller and less complex than a human brain, if we're talking about the kind of things which are going to break down in the case of neurodegenerative disease, like inhibiting the proper functioning of axons, or however messages get transmitted between neurons. I'm not an expert on this stuff, but obviously that's also going to be the sort of thing that could break down in a fly as well, because if you're talking about just things like movements which are controlled by anything that's controlled by a brain, I guess.
No, there's always the ones that we study have very obvious results in the fly, and some of these diseases actually you only get embryo stage, so the flies don't even develop anymore because it's such a severe thing on their neurons that they don't survive more than a tiny bean. That's what they look like on the microscope.
Right. This is what we were talking about earlier, the larva.
And then the larva is there's a lot of studies with larvae. Yes, there's for example, there's this is a cool one in my lab. There was someone doing epilepsy studies with larvae, where there's this basically what you can do is that you shock the larva and then you see how long it takes for it to start moving again. Again, if you were to do that to a human, maybe you wouldn't get the ethics approval that you would need to just go around and shocking people and then just time how long it takes them to recover. But that's one of the things that you can study. So basically these larvae had the genetic disease, and we were like, okay, one of the symptoms is seizures. So we are going to zap the thing and see how it goes. And I'm doing gestures, but they are actually microscopic, and everything that you need to do through a microscope, which is sort of like playing a video game. I've learned that playing video games gives you great microscopy skills when you need to dissect stuff.
That's good to know.
Yes, that was the one thing, because I did some experimental biology, that was the one thing that I was not terrible, which was due to years of video gaming. Just so you know,
the
kids.
Only real world application of being good with a video game controller would be being a drone pilot or something like that. But
also
microscopy. There you go.
Microscopy. Some of it, not all of it. It's very complex. I'm just talking about very basic.
Lava.
I'm just talking. Yeah, just that. On the other hand, if you have coffee, just think about this. This is a good thing for everyone to have a mental picture of how small these things are. If you have coffee, you're gonna have a slight shake on your hand, which is not noticeable for the entire of your day, but it's extremely noticeable when you're trying to pinch a larva or to pinch something. So when I did these experiments, I couldn't have any coffee, because just one coffee, the little amount, and I have a lot of caffeine per day, so technically it shouldn't affect me, but just the slight hint was so shaky that I would just smash and accidentally kill too many of them.
So you've got to be great at video games, but not a coffee drinker if you want to be good on a microscope, is what you're saying.
Or just tell everyone to not talk to you until you have the first coffee and be really upset the entire time that you're doing the experiment, which was what was happening. People would say good morning, and I was like, not today, and just walk past. And there's a reason why I'm not allowed in the lab anymore.
Well, on that fly torturing note, let me try and ask a conclusional question. Conclusional, is that a word? Let me ask a concluding question. Where do you see the research field of neurodegenerative disease going in the next few years? Is there going to be huge leaps and bounds made in how we think about curing these diseases, or is it still quite early days?
Yeah, this is more of a philosophical question at this point. So there's so little certainties in this field. So little. Like, there was things that we thought that were basic about Alzheimer's, which is the disease that is extensively researched. It's a hot topic in neurodegenerative diseases. There was things that we were given as something that was very important for the disease, and then it turns out it might not be that much, might not have the weight that we thought that it did. So the truth is that these are such complex things that it's very difficult to actually see where it's going. But for example, a few years ago, something very exciting came out, which helped a lot, which was, for example, technology is amazing. There was the development of a protocol that allows you to have brain structures in a dish. So you know how you can grow skin grafts in a dish from stem cells? That was a big thing that came out. Yeah, so there's a protocol to actually build these brain-like structures in a dish, so a 3D model of that, which is really cool and a massive technological advance. And before people say, but is that a mini brain? Does it think? No, that's why I'm saying brain-like structures in a dish.
But it sounds like you might be giving the flies a break, at least, potentially.
Well, they are having a break currently because there's no experiments going as of today because of the thing.
Lucky them.
But the flies are going to continue to be, because really they are a very, very powerful tool. Because if we can understand the basic mechanisms in this simple model, then we can extrapolate and try to understand the more complex. And as I said, it's just a really powerful and very exploitable tool. And also very easy to get. Like, people don't care if you kill 1,000 flies, but you go around and kill 1,000 humans, and suddenly you're a homicidal maniac. So that is I guess 1,000 you're a genocidal maniac, but you get the point.
Yeah,
this is a good conclusion, isn't it?
No one cares about flies. Those poor flies. I'm gonna start a fly advocacy rights organisation now.
You're just gonna go, invertebrate lives matter too.
They do, exactly, yeah. Well, on that note, again, fly killing. Let me thank you for joining or coming onto the podcast, B. It's been great having you.
Oh, thank you for having me.
No problem. And it's been really interesting look at your research into neurodegenerative disease. Best of luck with your write-up of your thesis. Have you got what percentage of the way through are you?
Oh God, that is not something that you ask a PhD student. That is in the writing up stage. I will now end this and go cry in the corner. This is what you do to me. No, in all seriousness,
if people are interested, B.
Yeah.
What were you going to say?
I was just going to say that, in all honesty, it is being written, so that's already a positive thing. So we'll see how it goes.
That's good to hear. Where can people contact you online? Are you on Twitter, or do you have a website or anything?
Yes, I do. I have one nickname that is on all social media, which is Moonian, and that is MOONIEAN. That took me a second, sorry. But I have a GitHub page, and I have Twitter, and I have Instagram, and I do occasionally stand-up comedy, and that's why I am also quite present online, because I'm trying to convince people that I'm funny, but it's just a very difficult task.
And if I remember rightly, is it science-related comedy?
Yes, I actually will be doing science communication competition this Friday. That's gonna be wonderful.
I would get you to plug it, but this episode will probably be
no, it's not gonna. But on the other hand, I do have videos about science-related comedy on YouTube that people can access from my Twitter. I also have some not so safe for work comedy skits, but those I can't plug here. But
well, we'll leave our listeners to discover those in their own time.
It's just like, right. Yes.
Thanks very much for coming on Podcast B.
Thank you.
To learn more about the work going on at the Alan Turing Institute, visit our website at turing.ac.uk. To get in touch with the podcast team if you have any questions or suggestions, email us at podcast@turing.ac.uk.
Music for this episode was provided by Jamin Sun. You can listen to his latest releases at jaminsun.bandcamp.com.
The Turing Podcast is hosted by Ed Calstry, Tara Callum, Ben Walden, Effie Dennis, and produced by Dan Whitfield for the Alan Turing Institute.